Good morning, everyone. We have a real treat today. It is my great pleasure to introduce our visiting professor today at Medical Grand Rounds, at Cardiovascular Grand Rounds. Dr. DEEPAK SRIVASTAVA is the director of the Gladstone Institute for Cardiovascular Disease. He also has an endowed professorship at UCSF in pediatric cardiology. He is a fellow Texan. He did his undergrad at Rice and then went from there to do his medical school training at UTMB Galveston, and then UCSF, right, for residency, and then Southwestern for, oh, you went to Children's in Boston. You bounced around a little bit, but ultimately ended up at UT as a faculty member before you went to the Gladstone, where he now directs the Institute for Cardiovascular Disease. DEEPAK has studied cardiovascular development, and he has made major contributions to our understanding of the determinants of cell fate. He's looked at chamber-specific transcriptional gene expression, and he has used that knowledge to manipulate cell fate. He was one of the first, if not the first, to use microRNA in mammalian systems, study microRNA in mammalian systems to understand gene networks in the heart that regulate cell fate. And with that knowledge, he was able to be the first to change cardiac fibroblasts, transdifferentiate those into cardiomyocytes, first in vitro and then in vivo, with an improvement in the MI model that he uses. So today, he's going to expand on his insights in reprogramming of the cardiovasculature, and we're going to hear from him in just a moment. Thank you, DEEPAK, for joining us. Why don't you join me up here, and we'll get started.
Hey, thank you. Great. Thank you, John. It's always a pleasure to be back here in my hometown in Houston. And I have to say that, as you can imagine, I was very keenly watching the news the last few weeks after the hurricane hit, and I was really affected by all the trouble that was going on here. But then I was so proud of seeing how the Texas can-do attitude kicked in, and to see how Houston just really responded to the hurricane. And coming here yesterday, seeing how and hearing more about how the medical center pulled together to deal with this was really impressive. So congratulations to all of you, and I'm delighted to be here today. Thank you for having me. As John said, our laboratory has really focused on trying to understand how cells acquire their fate, and then how we can use that understanding to address human disease. And so the themes that I'll share with you today are indicated here. I won't talk so much about our work on understanding gene networks that dictate cell fate, but rather how we might apply that. But in a nutshell, we have over the years figured out the major signaling pathways, transcriptional networks, and translational networks through microRNAs that are telling an early cell in an embryo that you should become a heart cell. And we have also then asked, are those the gene networks that are disrupted in the setting of human disease, and have identified several human disease genes, and have used cellular reprogramming approaches to understand mechanisms and find new drug discovery for certain diseases. And I'll share some of that work with you with some vignettes. And then finally, I'll end with what Jonathan mentioned, is how we can use that same developmental biology knowledge to regulate cell fate in the adult for regenerative medicine. And so all of the work that I'll share with you starts with that first bullet, which is just deeply understanding what are those gene networks that are dictating cell fate. Because if you don't understand that, then you don't really have the roadmap by which to either control fate or understand how disease might perturb such a network and shift it ever so slightly, resulting in the manifestations that we call disease. But if you do understand that, then you know how to shift that network back to some better state of homeostasis, and that's really our objective. And so having understood some of these gene networks, which are displayed on this slide, a subset of those, we can begin to assemble the key genes in some hierarchical networks, like what you see here. And what you're seeing here is what a heart looks like about 5 weeks of gestation in the human, where a tube is formed and it's looped, and you don't have walls yet separating, say, the right and left ventricle, but the cells that are going to become those chambers are very much already specified. In work I won't show you, we've now, with the latest technology, been able to analyze cells during a developing embryo at the single cell level, and even at about 3 weeks after fertilization, or even before that, when the heart isn't even a tube that beats, but it's a crescent, the cells at that stage already know exactly what they're going to be, whether they're going to be here, 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 or here in the atria. It's already specified. It's really unexpected, and we didn't expect it to be that early. Nevertheless, we can assemble hierarchies of genes like you see here. These are all transcriptional regulators, meaning these are proteins that will sit on DNA and turn hundreds, if not thousands, of genes on and off. So they serve as sort of the switches of the genome to turn things on and off. And what I've indicated here by asterisk are those genes that we now know, when mutated in humans, actually cause disease. And so you can see each of these networks are hit one or more times. And so it's been satisfying that as we figured out the developmental biology and merged that with the human genetics, that in fact these were the major networks that are controlling human disease. And our laboratory had contributed to this body of literature by identifying families that had mutations in this gene called Notch1, which is a very important developmental transcription factor I'll tell you a bit more about in a moment, as well as a protein called GATA4, which is involved in septation, and this gene was involved in aortic valve homeostasis. So we figured this out actually a long time ago, even when I was in Texas, and reported these families, but then got stuck for a while. And it was largely because the mouse model, which we tried to understand mechanism in, just didn't do the trick for us. And it's not enough to just know that this gene causes a disease in humans. If that's as far as you get, the chances of then being able to utilize that in any way clinically is small. You have to understand the mechanism in order to understand how to correct it. And so that's what I want to share with you in the next part. For both of those genes, I'll share with you two slightly different stories that come at them from different angles. The first relates to this gene Notch1, and it took about 10 years before we really figured out the mechanism, and that was work done by this brilliant MD PhD student, Christina Theodoris, who is now a pediatric resident at Children's in Boston. And this is the family that she had been trying to study that we reported some years ago. So you see here, this is a multi-generational family where those in the black are the ones affected by disease across many generations. The disease they had was that most, but not all, were born with a bicuspid aortic valve, which, as you all know, occurs in 1% of the population, most common birth defect that occurs of all. And most people, when they're born with a bicuspid aortic valve, like what it looks like here, instead of the normal 3-leaflet valve, don't even know they have it when they're born. As they get older, in an age-dependent fashion, in about 1/3 of those individuals, the valve calcifies, like you see here in this cartoon, and they then need a aortic valve replacement. And there are about 100,000 of those done a year in the United States alone. And we haven't known the genetic cause for this at all, and I think this remains one of the only known genetic causes. But the people in this family with 100% penetrance were heterozygous for a Notch mutation. So just one copy is gone. It was a loss of function, so they have 50% dosage. So the key question is, why does this occur? We did know, and those of you who are clinicians in the audience know this, that when people get aortic valve calcification, typically the leaflet, if you zoom in on this being the valve region, this is a leaflet, the leaflet exposed to this this laminar flow from the ventricle out is generally more protected from calcification than the backside of the leaflet, which is exposed to these eddy currents back here, and that's actually where most of the calcification occurs. So we had the notion that shear stress is somehow protective for calcification, but again, we didn't really know why. So the real breakthrough in this, because as I mentioned, we made mice that were heterozygous for Notch, and they were completely fine. They didn't care at all. If you bred them to homozygosity, they died as early embryos because it's essential for all vascular development. So the real breakthrough came with the discovery of one of my colleagues at Gladstone, Shinya Yamanaka, who made the transformative observation that one could take a cell from an adult, any one of you, your skin fibroblast or your blood cell, and put in just a combination of 4 critical genes that were coding for transcription factors, these switches, and those 4 factors were enough to reprogram those skin fibroblasts all the way back to a cell that behaved just like a human embryonic stem cell in virtually every way we can measure. So this was a real revolutionary discovery. It obviated the whole ethical debate around human embryonic stem cells, because these were just like human embryonic stem cells but didn't require destruction of an embryo. So it was a technology everybody could get behind. And it fundamentally changed the way we thought about the permanence of an adult cell and raised the issue that, in fact, those cells could be plastic enough to alter their fate if you knew how to do it. And I'll come back to that in a little bit. But these cells that now he called induced pluripotent stem cells, or IPS cells for short, obviously could be used by turning them into the cell type that's been lost in some human disease and then transplanting them back into that patient. And now these would be genetically identical, so maybe you wouldn't need immunosuppression for rejection reasons. And so that sort of approach is being pursued by many. And I think there's a lot of hope there. And it's already been in clinical trials for macular degeneration. And I think there's a lot of promise for a lot of diseases for this transplantation approach. But the other approach that I'll tell you about that is relevant to the genetics that I'm telling you about is to be able to use these cells to turn them into the cell type affected in human disease, and then not transplant those, but use them in a dish to understand the mechanism of disease, say in a genetically defined disorder. And if you can do that, then you can not only understand the mechanism, then you can use those cells to now screen for drugs that would alter that phenotype. So now you're looking for drugs for a human disease on a human cell of the exact same type that's affected in that patient. So it still may not work at the end of the day because humans are complex, but the odds of that working when you go to translate that and do clinical trials may be a lot higher if you've started off with the most relevant cell population. So this is what the approach that Christina took by making IPS cells from about 4 members of that family I showed you with the mutation, 4 members without, and then exposing them to shear stress or not to look at that difference of how that affects these cells. And in this case, differentiating them to endothelial cells, because from other work that I won't have time to share with you, we had concluded that it's the endothelial lining of the valve that is the most important. And so in these cells, which now we can create in billions, so she probably made 2 to 3 billion cells, endothelial cells, to do this project, she could ask, where does this Notch protein actually sit on DNA, because that would tell you what it might turn on and off. How does it affect the epigenetic landscape at those sites across the whole genome, and how does that then translate into transcriptional changes of the messenger RNA that might lead to disease. And I'm not going to go through all the details of this, but this is a tremendous amount of work, and then one has to computationally integrate these data sets, which itself is a computational hurdle. And the end result I'm just going to show you here in this cartoon. This is what we were able to conclude. And that is that Notch, what I didn't tell you, is not just a transcription factor that sits in the nucleus and attaches the genome to turn things on and off, but it's a unique type of transcription factor that actually starts off at the cell surface on the membrane. So it sticks up on the membrane and then when activated goes into the nucleus and affects gene transcription. But by virtue of being on the cell surface, you can imagine it's in a pretty good position to work as that sensor maybe of shear stress because it is where located here. And that in fact is what we found, is that just by virtue of liquid flowing across these endothelial cells, notch signaling gets activated normally. And then when it's activated, it goes in the nucleus, and its job in the valve is to actively suppress a gene program of calcification. And it does so by a variety of turning on anti-calcific genes that then prevent osteogenesis and inflammatory pathways. And in these patient cells, what's happening is when you have less notch, you end up de-repressing the system. You get upregulation of a few factors here that are all pro-osteogenic, and now the cells just get confused. They think they are supposed to be bone cells. So it's really a cell fate switch that's occurring in when you decrease notch. These cells, endothelial cells, begin to think that they should be osteoblasts, and they do what a good osteoblast should do, which is lay down calcium. So the crux of this disease of calcification in the valve, and maybe calcification in vessels as well, is that there's a cell fate switch that occurs where a cell starts to turn on aberrantly osteoblast-like genes. So that's great. It's useful to know. But what can we do about this? Well, it turns out that when Christina did deep network analysis, even though about 1,000 genes were dysregulated in this setting that we think are leading to disease, those 1,000 genes did not have 1,000 switches. It funneled down to 3 discrete transcription factors that I indicated here that get upregulated, and those 3 are responsible for about 80% of the genes that are dysregulated. The reason I share that with you is that then, if that's the case, then it becomes more conceivable how you could drug the system to target a more limited nodes that might affect the broad dysregulation. And so that gave us some hope that we could do a drug screen on these cells and maybe find something that would correct the problem. And so Christina did this in collaboration with another one of my colleagues at Gladstone, Sheng Ding, who's probably the world's leading chemist studying chemical biology, chemical regulation of fate. And so we screened about 1,500 drug compounds. And because Christina had mapped out all the genes that were dysregulated, she did a screen unlike any that had been done before, which is most drug screens look for 1 or 2 outputs that might give you a hint that, okay, maybe this drug is fixing a problem, and maybe 1 gene. What she did was and the technology, there's a company in the Bay Area that was developing this technology that we worked with, did direct RNA sequencing of 120 different genes that we knew were causing trouble at many nodes. And so for each drug, imagine we're assaying 120 parameters to tell if that drug is helping or not. And so that, again, it's a big computational challenge, but this is what an output of that can look like. And this is the simplest way to describe it, and I'll walk you through this real quick. This is a principal component analysis where the position of a dot is telling you what its state might be. And the normal cells are indicated in blue here. Each dot indicates a well with a group of cells exposed to a drug or not. So these are just normal cells. This is where you want to be if you're normal endothelial cells in this cube. Let's see if I can see here. In green here are what the Notch 1 heterozygous cells look like. So this is different from these blue areas. This is where you don't want to be. And then in red is that same cell exposed to a drug. And what you can see is that most of the drugs do nothing to the position of these cells. They're all right here with the mutant population. But what we call hits are those here that now you see a red cell, a red circle dot that is now clustering with the blue, which is normal. So these, by this computational analysis, are mutant cells that now, by virtue of exposure to one drug, have shifted over to a population that's more similar to the normal wild-type cell. Okay? So those are what it hits. And we have about 5 or 6 of these drug compounds that went through validation, and now are compounds that we'd like to try in some clinical setting. But of course, we're not going to go straight from these cells in a dish to a clinical trial. We'd really like to be able to test this in an animal. But you'll recall I mentioned earlier that the Notch1 heterozygous mice were totally fine. So we didn't have a really good place to go after we got these drugs to test if they could actually work in vivo. So Christina then made the following observation. As I mentioned earlier, that this is an age-dependent disease. So, as clinicians know, we'll often diagnose it when somebody's 40, 50, 60 years old. We'll do serial echoes, and when it gets bad enough, we'll replace it. We don't really have an intervention. But what we do know is that from explants of people who've had their valve replaced, these valves, the telomeres in calcified valves are actually shorter than the telomeres in age-matched valves. And you all know, I'm sure, that telomere length is somehow associated with aging, even though we don't know exactly how. And then the other observation that she made is that mice actually have much longer telomeres normally than humans. And so the hypothesis that then all this leads to is that could it be that the reason Notch1 heterozygous mice don't get calcification is that their longer telomeres actually protect them from the manifestation of this disease. So that is a testable hypothesis. And we can do that, and Christina did this, by taking mice that were heterozygous for Notch and crossing them with mice that were mutant for something called TERK, which is the enzymatic component of telomerase. So now you get, as you breed these mice, in each generation you get shorter and shorter telomeres because they can't restore their telomere length. So we did this, and this is what I'll show you in the next slide is really a remarkable result by just the second generation, which is not shortening a whole lot. We were able to completely recapitulate the human disease, and it very specifically to the aortic valve rather than any other problems, even though Notch, actually, I didn't mention, is important in development of virtually every organ in your body. And so this is what it looked like. So here's a cross-section of an aortic valve. These are the leaflets. You see nice and thin. No calcification when stained with alizarin red, which is a marker for calcium deposition, and at high magnification. In contrast, here's the mice with shorter telomeres. This is the aortic valve leaflet here. You can see all this calcification here. If you zoom in, this is really extensive. And by echo, we can even pick up a gradient across the valve suggestive of true aortic valve stenosis. And so we have these sections. We went a step further and stained these sections for a protein called RUNX2. And the reason is that RUNX2 is another one of these key cell fate determining transcription factors that's the major switch for an osteoblast. So if you put RUNX2 into a cell that's not an osteoblast, it starts to become an osteoblast. And so you can see here in green that these cells that are getting calcified are all expressing RUNX2 where they normally shouldn't. And so this is consistent with the notion that I described to you earlier, where we think these cells are undergoing a cell fate switch and are now thinking that they're bone cells. And so one other observation we made from this that was very interesting for me, particularly as a pediatric cardiologist, is that if you shorten the telomeres a little bit, you get calcific aortic valve disease. If you shorten them a bit further, we ended up getting, if you zoom it, look in here, this is the left ventricle, this is the aorta, here's the aortic valve. These are thick, big leaflets instead of the thin ones I showed you they're supposed to be earlier. And this is in a newborn mouse. And so this mouse actually developed critical aortic valve stenosis, died right after birth. And the reason I bring this up is that I didn't mention earlier, in the family I showed you initially, and what we see clinically, is that within the same family, there'll be some people who are 60 years old and get calcific aortic valve disease, and that's the first they know they've ever had a valve problem. And in the same family, there'll be a kid who's born and needs a balloon aortic valve dilation in the first week of life. So clearly, these two aortic valve diseases are somehow genetically connected, but we've never known exactly how. And we think that in what we're seeing here, even in the same genetic background, just by changing the length of the telomeres, you're getting manifestation of the whole spectrum of aortic valve disease. So now we're going back to some of our patient population and asking, could that heterogeneity of manifestation of disease within families be attributed to telomere length? And we don't know the answer to that yet, but I think that's what the hypothesis this would lead to. In any case, now we have a mouse model where we can test the drugs that we identified from the human iPS cells, and now we're doing those preclinical trials in mice, and we don't have the answers yet, but I'm very hopeful that at least one of those may alter the natural history. And again, as you can imagine, it being an age-dependent disease, we don't have to stop this disease. We just need to slow it down, and people will, end up succumbing to something else. So if you can just shift the curve so you don't need to intervene in a 70-year-old and can shift that out 10 years, that's probably enough to make a significant clinical difference in this population. So that's where we're headed. We're also testing these drugs on a panel of other human iPS lines we've made from people without notch mutations, just garden variety calcific aortic valve disease, so we can determine in a dish, sort of in a clinical trial in a dish, who might be responders and who might not, and that might inform the selection of patients for a subsequent clinical trial down the road. So that story I share with you to give you an example of how one can go from human genetics and understanding a genetic cause of a disease to understanding mechanisms using human-induced pluripotent stem cells, all the way to drug discovery and testing in animals. But you can imagine if we didn't have all those pieces, we couldn't do any of this. And all this has really come of age only in the last few years. The next story I want to tell you is somewhat related, but I want to get at what I didn't talk about there, which is the developmental abnormality. So you'll notice that I didn't say a word about why these patients got bicuspid aortic valve from a developmental standpoint, frankly, because we haven't been able to figure that out. But I do want to share with you, I think, how we figured out a morphologic developmental defect in this family. And this is another family we reported a long time ago that, again, many generations, there are about 20 people here affected. They've continued to have kids. Now they have almost 30 people in this family who have had atrial septal defects and ventricular septal defects. We had mapped the critical mutation to a heterozygous missense mutation, so just one amino acid's changed on one allele, but that was enough in this protein GATA4 to cause disease. So again, the question is why? So what we have found out that this point mutation, this single amino acid, ends up disrupting a very key interaction with the second factor called TBX5, which is the disease gene for Holt-Oram syndrome, which also causes ASDs and VSDs. And this mutation sits right next to a part of the protein called a zinc finger that's necessary for these protein interactions. And so when we went to make iPS cells from this family, we did so with this branch of the family. All these kids here had septal defects, the parent did. So we made iPS from these controls and these diseased folks. So this was several years after we first reported them. We moved to San Francisco. We had the family fly up to San Francisco. And we thought, OK, the kids are older now. We should do echoes on them just to see where they stand. We had a surprise then. So this is obviously what a normal echo looks like. Here's a right ventricle, left ventricle. You have these nice thin walls here. And this is what it turned out. The kids who were now teenagers, when we did their echoes, they had cardiac dysfunction. Their holes had been closed. And normally if you have an ASD or VSD, our surgeons are great at closing them, and they're fine for life. They should not cause any long-term problems. Here, where you see these deep trabeculations in the right ventricle that had developed, sort of like ventricular non-compaction, which we typically see on the left ventricle, but is here in the right ventricle, and then this very thick papillary muscle, and then some dysfunction. So this was a total surprise, but it did give us some hope that, well, if we can't figure out why they had holes in their heart, which we thought maybe it would be difficult on a two-dimensional dish with a bunch of beating cardiomyocytes, maybe we could at least figure this out, because this has been thought to be a disease of immaturity, if you will, of cardiomyocytes. And so Jensen Ang, a really smart postdoc in the lab, and Renee Rivas, an MD PhD student who's actually from Texas, from Dallas, did this work. And we reported this last year, and I'll just show you the highlights. When we made iPS cells now from these, we had a new technology that we didn't have before called CRISPR gene editing that very efficiently allows us in the human genome to go in and with a genetic scalpel almost, correct or change a single nucleotide. And so that allowed us to do that, to change this mutant form back to normal. So we'd have what we call isogenic controls, which is the best way to do this. So you have everything in the genome's exactly the same in these cells except for one nucleotide. And so the question then is, could we model some decrease in contractility here? And we were able to do that by putting individual cardiomyocytes. So we made these iPS cells, turned them into beating cardiomyocytes, which we can do quite well, and put them in these patterned wells. So a single cardiomyocyte would form the way we wanted it to, and then we can measure the force that that single cardiomyocyte generates in that well. And so that's what this measurement looks like. So here's each dot represents one cell and the force it generates. This is a normal cell, and here are all the mutant cells. So what you can see, I think, is that even though we've gone from a skin cell of a patient to a stem cell and then turned it into a beating cardiomyocyte and gone through all that process, we're still able to model the contractility defect caused by this single gene mutation, which is really remarkable if you think about that. If nothing else, the fact that you're able to do that is really amazing. So then the deeper question is why? And so once again, we did something very similar to what I described earlier of asking, where does this factor sit on DNA, how does it affect the epigenetic landscape, and how does it affect the transcriptome? And once again, I'm just going to show you the take-home point for this without all the gory details. And that is, we found that in this case, these two proteins that I mentioned are obligatory cofactors for one another at thousands of sites across the genome. So they need to interact with each other to do their job. And their job is to activate high levels of cardiomyocyte gene expression. But it's not just that. What we found is these same two factors sit at hundreds of other loci in the genome to tell those areas to turn off. So it turns out that the same factors that are turning genes on are sitting at other places as like a binary switch and telling, okay, if you're going to become a cardiomyocyte, you don't want to turn on endothelial cell genes, you don't want to turn on smooth muscle cell genes, et cetera. So they're doing both. And in the setting of disease, you get not only downregulation of cardiomyocyte genes, but we saw broad upregulation of a number of other cell types genes, particularly endothelial genes. So these cells, they were cardiomyocytes, but they were confused cardiomyocytes and were more immature and thought that they were a little bit like an endothelial cell. And we think that is the crux of the problem here. But what I'm most excited about is that I think we were able to crack the issue of why do patients get holes in their heart with this mutation. And that's what I'm going to show you in the next few slides. So before I do that, I have to back up and give you a little bit of a primer on some developmental biology. And that is that as the heart is forming at this stage, before the walls have started forming, here's the ventricular septum, it's not fully formed. And it looks something like this. Here's the heart in a mouse embryo. Right behind this forming heart, in a very discrete location, is the pulmonary endoderm that's forming, the lung bud. And that pulmonary endoderm secretes a very critical morphogen called sonic hedgehog. So it just diffuses locally. So the only part of this myocardium that'll be in a position to receive this signal is right here at the crux of the heart where the septa will form. The rest of the myocytes, even if they're capable of having the machinery to receive this signal, are not going to receive it. So they don't care whether it's there or not, whether they have the machinery or not. So if you look at an embryo, here's the sonic hedgehog signal. It's right behind the heart. Here on a side view of the heart would be here, and here's the pulmonary endoderm. Okay. And we've known from other studies that this sonic hedgehog signal is critical to form the septum. And the reason that is we know that is that if you either delete sonic hedgehog specifically from the pulmonary endoderm, or you mutate the genes that are on the cell surface of the myocardium that are needed to receive the signal, then in both cases you end up with mice that don't have the atrial septum, which is right here on histology, and they just have a hole, a big atrial septal defect. So this signal is critical to be sent and to be received. And the way this pathway works is that the morphogen is received by a receptor called patched, transmitted by another cell membrane factor called smoothened, and then in the nucleus, a factor called GLE turns on and off genes. So this is the machinery in the myocyte that has to be there to receive the signal. Now, these are a lot of names that you may not have heard of. The names aren't important. What is important is that you have to have the signal. You have to have all these here to have the system respond correctly. So why am I telling you all this? Because when we looked at the transcriptome of the cells that didn't have this single amino acid changed in GATA4, we saw broad downregulation as indicated in this heat map, with blue being lower and red being higher gene levels of gene expression of a whole panel of genes that were essential for sonic hedgehog signaling. And in particular, you have things like patched 1 and 2. There are 2 forms of it, GLE 2 and 3. So all the machinery that's needed to receive the signal are downregulated in the setting of this mutation. So what that means is that as a heart is forming, the muscle cells just simply aren't capable of receiving this critical morphogen, and that may be why that in these patients, or in this situation, they don't form the proper septa. So again, that is a testable hypothesis, and this was work actually not done by our group, but subsequently by Ivan Moskowitz's group in Chicago, where they had come to this thought also by studying GATA4 heterozygous mice that do have septal defects. And they did the experiment where they crossed this mouse, you can see here a big atrial septal defect, with a mouse. This is the beauty of mouse genetics. You have all these tools. There's a mouse that has a mutation in that smoothen protein that makes it constitutively active, meaning it doesn't need to receive a sonic hedgehog signal. It's always receiving the downstream signal. So if you cross this mouse with this constitutively active form, then maybe you could rescue the holes in the heart, and that would prove that this, in fact, was the problem. And that's what they found. So in about 90% of the embryos in this setting, the atrial septal defect was rescued. And so this makes us think that, in fact, in the patients with this mutation, the reason they get septal defects is that even though all their myocardium is now in a position where they can't really receive the sonic hedgehog signal, most of it doesn't care. The only part that cares during development is that little bit that is supposed to receive the signal. And that's why we were able to model this. Even though we're studying only 2 dimensions, we could figure out the basis for this 3-dimensional disease. So this vignette I wanted to share with you because it illustrates how we can use this system to understand a birth defect. Okay, now finally, in the last bit, I'd like to turn our attention to this concept of, can we use these same developmental pathways that I've told you about so far and use them in a clever way for regenerative medicine? And here the issue is not the pediatric form of disease, but an adult form that you see here. And it doesn't really matter what form of heart failure you're talking about, whether it's caused by what you see here, the consequences of maybe an MI, where you've got scar tissue all throughout here in the left ventricle, or myocyte dysfunction, where you simply have progressive loss of myocytes. Any situation where you've got heart failure because you've lost myocytes, the ultimate treatment would be to regain myocytes. And we as a field have tried to do that in a variety of ways by injecting cardiomyocytes into this area from stem cells or other sources. But we've faced the hurdles of getting those cells to survive when we put them in, to stick and not get squirted out, essentially back in the circulation, to then electrically integrate with their neighbors, which has been challenging, and ultimately to improve function. And so many years ago, we recognized that given the difficulty we were having with that approach, that maybe we should try a different approach. And we knew that in the human heart, only half the cells are actually myocytes. The other half are fibroblasts. So you've got this abundant reservoir of cells in the organ sitting there that if you could somehow harness to your advantage, that maybe you could regenerate with those cells. And the reason we began to think this is that, as I mentioned, Shinya Yamanaka, who I didn't mention had won the Nobel Prize for this conversion of fibroblasts to stem cells, was down the hall from us. And we thought, well, if you can find the right code, hack the code so you could convert a fibroblast to a stem cell and then a cardiomyocyte? What if you could put in a different code, maybe the code for a myocyte, and convert that resident fibroblast right into a beating cardiomyocyte? And could do that in situ right there in the organ. And so we set out to try to do that, and we've published a body of work on this over the last few years, and I'll just summarize for you in a few slides that work. That is that we found that a combination of just 3 factors shown here, 2 of which I've already told you about, we now know are obligate partners to direct cardiac gene transcription, and a third, MEF2, together were sufficient in vitro to push cells from a fibroblast state closer to a cardiomyocyte state, somewhat inefficiently by the parameter of going all the way to a beating state, but could really shift their fate. We didn't just want to do this on plastic in addition and put them back in the heart. We wanted to do it directly in the heart. And so Li Qian, who has her own lab at UNC, did this work in our lab where she ligated the coronary artery in mice, used a gene therapy approach to inject these 3 genes in the myocardium, used mice where we could genetically label with a fluorescent marker the non-myocyte population. So we could ask, are those the cells that now become myocytes with nice sarcomeres, et cetera? And then what she found is that, in fact, we were able to get much better conversion in vivo than in vitro. These cells electrically coupled with one another, which was key to get improved function. And they're most similar to an adult ventricular cardiomyocyte. And by MRI, which is the most rigorous measure, they could improve function. And histologically, it looks something like this, where here is a cross-section to the left ventricle, and this is all the scar tissue here. And when we introduce these 3 genes, we still get thinning of the left ventricular wall, scar, but now we've got myocardium in this region. And if you use this fluorescent marker, these are all fluorescently positive, suggesting to us that these used to be non-myocytes that have now converted. Now, since this time when we reported this, many groups across the country have made improvements to this protocol and added various ways to make this more efficient, including Todd Rosengart here in the medical center, who's worked on this, and in adding VEGF into this in vivo can get even greater improvement. And I should say that the improvement we see by MRI we think is partly due to creation of new muscle, but the fibroblasts that don't convert their fate but have taken up these genes are intrinsically different, and they may lay down scar in a different way or collagen secretion. And so it's hard to parse out how much is being contributed by each, but I think it's safe to say that it's likely that both are contributing. Now, to make this more efficient, Tamer Mohammed, another postdoc in our lab, screened a chemical library of about 5,000 compounds with these factors to ask in vitro, can we make this even better in an unbiased fashion? And we recently reported this, but I'll share with you Tamer's findings, which is that normally in vitro, the process is fairly slow to convert a cell. And it takes about 6 weeks before cells actually beat in vitro. When he, out of that screen of 5,500 molecules, one of the hits was a TGF-beta inhibitor. And when we added this, we not only got more cells converting, but they converted more quickly. About 3 weeks, they started beating. And then a second hit from that was a Wnt inhibitor, another important signaling pathway. And remarkably, when we put those 2 together, within a week, we were starting to see cells that were beating in the dish, which really is amazing when you think about the epigenetic change that has to happen over the whole genomic landscape to take a cell from being a fibroblast all the way to a beating cell with sarcomeres. It's really amazing that this could happen so quickly, but that is what we can see quite efficiently. And I won't share with you the data, but we can map out the transcriptional changes that occur even at day 1, 2, 3, and 4 with this. And actually, most of the change occurs within 2 days. And then the cell catches up with its cell biology and makes its sarcomeres. And so we went back to the in vivo model and tested these, the genes that I mentioned earlier, with direct injection in the myocardium, but now with the 2-week systemic delivery of these 2 chemical modulators. And here's sections from at the apex of the heart and more at the base after 3 months. And here's all the scar you see at the apex, which is typical. And here, even at the apex of the heart, where we expect the most damage, with this combination, we're seeing quite a bit of muscle, and all this muscle that you see here is all fluorescently marked with this non-myocyte marker. So all this is new myocardium, and as you go higher up, there's a lot of myocardium. So we think that this combination of genes and chemical inhibitors is probably the most promising approach. If you look by functional analysis, by ejection fraction, here by echo, MRI is very similar, and look at the decline in EF over time. These are the controls. This is with the genes alone, and then here is with the genes plus chemicals, and you can see that there's a significant improvement over what we were doing before with this combination. So that's great, but obviously we don't want to just treat mice, we want to treat humans. And it turned out that this combination of genes that worked well in mice actually didn't do much in humans at all. So we had to go back, and it turned out to be a more complicated combination of 5 or 6 genes that really worked in humans, which then gets to be an unwieldy amount of genetic material. So we went back and asked, could these chemicals also help the human situation and maybe simplify the cocktail? And fortunately, that turned out to be the case. And that is that with these chemical inhibitors, it turns out that a slightly different cocktail shown here with other transcription factors were enough to actually make these nice sarcomeres in a human cardiac fibroblast. And so now we've compacted the amount of material and work I won't show you. We've tried this in pigs in a size that's more similar to humans. We can get some improvement there in our preliminary studies. But then the challenge comes, okay, how do you package this material for gene therapy that it could actually be useful? And we've thought that the integrating kind of vectors, retrovirus, lentivirus, may not be so clinically applicable. An AAV might be ideal, adeno-associated virus. But then we ran into the hurdle that if you wanted to use AAV to deliver a gene to cardiomyocytes, we actually have some great AAVs that have good specificity, and that would be fine. Obviously, that's not our target here. We want to deliver to the fibroblast, and it turns out there really weren't any good fibroblast AAVs that could efficiently infect those. So we spent several years then screening an error-prone library in collaboration with David Schaefer's lab, where you have millions of different AAV capsids through random mutation, and you can screen that space for one that would infect a human cardiac fibroblast. So we did that. We found one. We packaged this stuff into that one, and I just want to show you the recent results with that. If I can get my arrow to play this video, that in fact, with this AAV, we can get high enough levels of delivery into a human fibroblast, and here's a beautiful beating cell that we're seeing just a week after introducing these into cardiac fibroblast. So we've taken this pretty far in our hands, a bit out of our comfort zone from the translational space. I'm pleased to say that very recently, less than a year ago, we've been able to spin out a company and get commercial investment with a $50 million Series A financing to license this technology and take it towards a clinical trial with Dave Goodell and the Column Group. So that I'll stay involved, but I think we need somebody doing this who knows what they're doing. And so they'll pursue that. We are also collaborating with the group here, with John's group, to try to get messenger RNA, modified mRNA, as a delivery vehicle instead of the viral vectors. If we can get those, now that we have these chemicals that switch the fate so quickly, it's conceivable to think that if we could get 4 or 5 days of protein expression from the modified RNA in vivo, that may be enough to use modified RNA, and there's one of the country's best cores here to make these, and they're making those for us right now, and we're eager to test those for this process. So in closing, what I hope I've shown you is that by simply understanding deeply the gene networks that control cardiac cell fate and understanding how those networks are perturbed in the setting of human disease, we finally have the opportunity to actually try to make a difference in many human conditions by altering these gene networks to our favor for various diseases. And so I'm very hopeful about the future, and I'm grateful to the people on this slide who are in the lab right now, who are really a wonderful, dedicated group of folks, and a number of former lab members that I tried to acknowledge as I went along, some of whom have gone to that startup company, and then we've had wonderful collaborators over the years that are shown here. And with that, I'll stop and be happy to take some questions.
Thank you, Deepak. That was spectacular, all the way from molecule to man. And really, you've done so much for our understanding of cell fate in the heart, and it's wonderful to see that it's making its way to the clinic now. I have a question about the reprogramming of cardiac fibroblasts. I think all of us that are doing reprogramming notice that in a field of fibroblasts, there are actually very few fibroblasts that reprogram. My question to you is, what explains that heterogeneity of response to the reprogramming factors? Are there some differences in the fibroblasts that are responsible for some being more readily reprogrammed? Mm-hmm. And then what does that say when you apply this technology to the heart in vivo?
Yeah, those are really good points. And what the issue is that even in the best of circumstances, probably we're reprogramming 15 to 20% of fibroblasts. Sometimes we get up to 30, but still only a subset are switching their fate. And why is that? Is that some are more amenable than others? And so we've only recently been able to finally tackle that with the technology that's come online in the last year involving single-cell RNA sequencing and doing that at scale. So just in the last year, 10x Genomics platform allows us to get whole RNA sequencing done on thousands of cells. And actually, I didn't have time. I actually took out this slide at the very end, which is the output of that. So here are about 50,000 cells. Each dot is a single cell that we can get the whole transcriptome on. We could never even dream of doing this a year and a half ago. And that allows us then to begin to ask the question you raised, is what is each cell doing in the dish? And so I won't go through all this in detail because there's a lot of information here, but these are cells during the reprogramming process right before reprogramming, a day after, 2 days after, 3, et cetera. So we can map out every day with and without chemicals. These are the fibroblast population that we start with. These are the myocytes where they end up. And so what we can tell is that they go on this arc in this principal component analysis pattern. And I won't go through why we know this, but from this, what it appears to be happening is that within a day or two, many cells that get infected with these factors go off on a tangent. Some become blood-like, some become just weird cells that are not fibroblasts or myocytes, and only a fraction go down a path, but that path is determined within a day or two, and they go to this area, and then they're on a path to end up here over time, and the rest end up over here. We are interrogating why that is now that we've got a handle, but I think it's largely the stoichiometry of what the cells get and how much they get. And then there's some stochastic events that must be there that just a cell, maybe where it is in its cell cycle, goes off on a different path. So we have the ability to interrogate that, and we're in the middle of doing that.
Thank you for that. It's fascinating work. I think we're privileged to have all that information shared. My question is a little more fundamental conceptually in cell fate and reprogramming. When a cell takes up a fate, as you've shown very early, and whether you use Yamanaka factors or there's a natural reprogramming, is that a switch on and it's a one-way street? Or do you have to keep it in that by hormonal factors? And is there a fate elasticity that they're always trying to go back to that fate?
That's a great question. I didn't mention that, but we have studied the epigenetics of that process, and it appears that up to a certain point in time, you need constant cues, and then it's self-sustaining. In IPS reprogramming, that's been true because we can use inducible genes, and the same in the cardiac where you can turn them on and off. And in our initial work for cardiac reprogramming, this is without the chemicals. After about 2 weeks, we could turn the genes off, and it's self-sustaining. It's much shorter with these, but we haven't nailed down exactly how many days, but my guess is it's within 5 or 6 days. So we don't need ongoing expression. Yeah.
So my question is about the myocardial infarction model. So normally inflammatory cells come in as a part of the healing after the infarction. So when you put the GMT, what was the role of the inflammatory cells in the process of this differentiation into cardiac myocytes? And if there is an influence, would it be better, yes, it's interesting to do the single cell in vitro, but would it also be interesting to know when you have different types of cells, what happens to the fate of individual fibroblasts versus others?
It's a great point, and it's something we'd very much like to do. The in vivo, as I mentioned, is more efficient and better quality, and so why is that? It's possible that many of those cues from the endogenous environment and potentially during the inflammatory condition are promoting that. We can get reprogramming even without injury, so it doesn't require inflammatory signals, and that's what the in vitro work would suggest, but it could be making it better. So we have designed now vectors that would allow us to isolate cells from in vivo reprogramming at the single cell level, just like we did in vitro. To get at exactly at your question, I think that'd be very interesting, and it might tell us what those cues are, which we don't know yet.
Well, we've reached the end of the hour.
Great.
Let's all thank DEEPAK for a great talk.
Thank you very much.